Hello, I'm Les Recio, the VP of Research Development at ILS, and today I'll be talking about the ILS genetic toxicology testing strategy to meet the ICH S2R1 compliance for pharmaceutical agents. The ICH S2R1 document gives the pharmaceutical companies two options for genetic toxicology test battery. One is the traditional approach, which includes a bacterial mutation assay, followed by an in vitro assessment of genotoxicity or chromosomal damage using mammalian cells. And a third test would be an in vivo assessment of chromosomal damage. There is a second option in ICH S2R1, which allows the conduct of the bacterial mutation assay, then going directly to the animal model using two genetic toxicology endpoints. One is chromosomal damage using the micronucleus assay, and secondly is an assessment of DNA damage in the liver. For these tests, ILS uses criteria to determine a positive or negative response as outlined in these documents. The ILS criteria is basically statistical tests for a positive response using a p less than 0.05 statistical significance, the presence or absence of a dose response, and finally, whether those data are within our 95% confidence interval of our historical control. If all these three criteria are met, the compound is positive. If none of these are met, the compound is negative. If only one or two of these are met, the compound is considered to be equivocal. In our case, we consider these to be not biologically relevant. At ILS, we conduct a number of approaches for this to meet this ICH S2R1 requirements. These include screening assays, which include computational models as well as biological models that employ miniaturized versions of the traditional definitive assays. We also employ the traditional GLP-compliant, OECD-compliant genetic toxicology test battery. In ILS, our in vitro mammalian cell genotoxicity assessment is preferred to be the human TK6 cell assay using micronucleus assay determined by flow cytometry. We use human TK6 cells because they are P53 competent and they are human relevant cell line. We use flow cytometry-based assessments because they provide an unbiased assessment of chromosomal damage, and they also have very high accuracy, eliminating user bias. We employ the flow cytometry-based micronucleus assay both in vitro and in vivo. And finally, for follow-up studies, we employ a number of traditional and investigational approaches. For instance, we use human hepatocyte models, including HEPA-RG, and gene expression profiling that assesses mode of action and potential involvement of a DNA damage response indicative of a genotoxic hazard. For the genetic toxicology screening assays, we use Case Ultra as our initial computational test. Case Ultra 2 is a software that's been supported by the US FDA. It is compliant with ICH M7, and the software we use gives you both the statistical assessment and the expert predictor. In a follow-up test for these, if it comes out positive in these tests, we typically would suggest stopping at this point or going on, if in the case of a negative, going on to do either the mini AIMS or the full GLP compliant AIMS test. The ILS Mini AIMS is a non-GLP test requiring less than 50 milligrams of compound. It uses three tester strains, two salmonella tester strains and the E. coli tester strain with PKM 101. This assay is a 6-well version of the full 100 millimeter plate version of the AIMS test. And in case of positive here, we would suggest you would stop here and move on to the next compound in a series of compounds. Or if it's negative, you go forward with in our case, we recommend doing an in vitro micronucleus assay, screening assay, which in ILS employs human TK6 cells. And in this case, we use flow cytometry-based assessment of cytotoxicity and flow cytometry assessment of micronucleus. Again, this is a low sample requirement assay, less than 100 milligrams of compound. And again, we use TK6 cells, which are P53 proficient. In case of a positive here, we would recommend stopping However, if it's negative, we would suggest going forward with the genetic toxicology test battery, which begins with the Ames test. The Ames test conducted at ILS is compliant with OECD 471. We use 4 salmonella tester strains and an E. coli strain that has been transfected with the PKM 101 plasmid. In the case of a positive here, you may want to stop here. However, in case of certain compounds, you may wish to go forward. In this case, you could either do a mammalian cell in vitro assessment of mutagenicity or the in vivo pig-A assay, which is a new assay developed for the assessment of mutagenicity in vivo by flow cytometry. In the case of a negative, you would of course go forward to the in vitro mammalian cell assessment of genotoxicity. And at ILS, we recommend the flow cytometry-based micronucleus assay in human TK6 cells. We use flow cytometry-based assay, and in this case, we use relative increase in cell counts determined manually to be compliant with OECD 487. At ILS, we also had the capability of using either CHO cells or peripheral blood lymphocytes in a micronucleus assay using traditional microscopy-based scoring. In the case of in vitro here, we would suggest going directly in vivo to the equivalent assessment of chromosomal damage or micronucleus assay in vivo with OECD 474. Or in the case of a positive, you may want to consider investigational studies in either metabolically competent HEPA-RG cells or human hepatocytes, or some mode of action assessment using gene expression profiling. If it's negative, you of course would go forward to the test battery. The other alternative chromosomal damage assessment is using traditional chromosomal aberration tests in either CHO cells or peripheral blood lymphocytes. These tests tend to have a higher rate of false positives in CHO cells. Human peripheral blood lymphocytes do not. However, neither one of these assays can detect aneuploidy, which is a significant concern. Also, the cost of these and time and effort has gone up significantly with the new requirement to count at least 300 metaphases per dose point. In the case of a positive here, we would still recommend going in vivo to the in vivo micronucleus test, or again, as I mentioned earlier for the micronucleus test, some metabolically competent human cell line or human hepatocyte model. In our case, we use HEPA-RGs. If it's negative, you go forward to the in vivo assessment of chromosomal damage using the in vivo micronucleus test. The in vivo micronucleus test conducted at ILS is compliant with OECD 474. It's a chromosomal damage assessment in both rats and mice. We prefer flow cytometry-based micronucleus assay in peripheral blood as our assay of choice. However, we do offer and have the capability of conducting slide-based micronucleus assessments in either the bone marrow or in the peripheral blood. In the case of a negative response here, you're basically finished with the test battery, or if it's positive, you may want to consider other alternatives that include genomic profiling to assess mode of action and a benchmark dose or a risk assessment. In addition to the in vivo micronucleus assay, ILS also conducts the in vivo comet assay. The comet assay we conducted at ILS is compliant with OECD 489, and it's an assessment of DNA damage typically in the liver or other tissues. The comet assay can be combined with the micronucleus assay in a combined test approach that we use at ILS. This saves animals and time and money and allows you to go forward at a quicker rate instead of going back and repeating each assay independently. In the case of a positive in the comet assay, we recommend histopathology assessments of those tissues because in the case of the comet assay, there's potential for false positives due to either cytotoxicity or apoptosis in that tissue. We also have recently demonstrated that tissues collected and cryopreserved for the comet assay can be used for gene expression profiling by next generation sequencing to provide mode of action or to assess the origin of that positive response in your assay. We also recommend in the case of a positive response in the comet assay to consider doing further studies that include and can include up to doing a CARC study. In case of a negative assay, you are finished with the test battery. So to conclude, ILS has a number of capabilities to meet the ICH S2R1 requirements. This includes screening assays that are both computational and biologically based models, also in the traditional ICH S21 genetic toxicology test battery. And finally, we also offer a number of follow-up studies that are investigational that include human hepatocytes, as well as HEPA-RG cells, and toxicogenomics-based gene expression profiling using state-of-the-art next-generation sequencing. Thank you very much for your time.